Hello dear students, welcome to this lecture series in botany. Today we are going to discuss an important topic, that is applications of plant tissue culture. In this lecture, following applications of tissue culture will be discussed: applications in horticulture and forestry, applications in plant breeding, and general applications. Plant tissue culture is applicable in the field of horticulture and forestry through micropropagation and production of disease-free plants. Micropropagation is the technique of producing plants from a very small portion of plant, which is known as explant. This explant can be a shoot tip, root tip, leaf portion, embryo, pollen grain, callus or single cell. Micropropagation is very important in horticulture and forestry. For example, it is applicable in regeneration of ornamentals. Micropropagation has a tremendous impact on ornamentals because of the high intrinsic value of the final product. Among ornamentals, orchids were the first horticultural plants that have been successfully micropropagated. Examples include Cymbidium, Dendrobium, Vanda, Phalaenopsis. Other ornamentals that have been regenerated in vitro include Cyclamen, Lilium, Dianthus and Pelargonium. Micropropagation is also useful to fruit and nut crops. Among fruits, more research has been done in case of apple, followed by peach, pear, plum, cherry, grapes, etc. Nuts include chestnut, walnut, pistachio, almond, etc. Micropropagation is important for conservation of medicinally important plants like Rauwolfia, Atropa, Dioscorea, aloe vera, curcuma, allium, etc. A number of woody plants have also been regenerated through the technique of micropropagation. Examples are ulmus, populus, eucalyptus, sequoia, pinus, betula, etc. A number of tropical and subtropical plants have also been regenerated via somatic embryogenesis. Examples include Carica, Phoenix, citrus, etc. Micropropagation has also led to the production of synthetic seeds. These synthetic seeds are produced by encapsulating the somatic embryos and then adding growth regulators and nutrients which acts as artificial medium. These synthetic seeds have proved helpful in regeneration of non-seed producing plants like banana and polyploid plants with elite traits like cardamom and rice. Micropropagation has also given propagules like mini-tubers and mini-corms, which can be used for the plant multiplication throughout the year irrespective of the season. Examples are Lilium and Gladiolus. Another importance of micropropagation in the field of horticulture and forestry is production of disease-free plants. There are three ways to eliminate pathogens from the plants. These are meristem tip culture, heat treatment and chemical treatment. Morel and Martin were the first persons who in 1952 applied tissue culture techniques for the production of disease-free plants from infected dahlia by culturing its meristem tips, because meristem tips, they are either free of pathogens or they contain very low concentration of it. Heat treatment is given either through hot water or hot air. Hot water treatment is recommended for dormant buds, while as hot air treatment is recommended for actively growing shoots. Example where heat treatment has proved successful is in case of Dianthus caryophyllus, where all viruses from its shoot tips were eradicated by continuous treatment of plants at 38 degrees centigrade for 2 months. Another example is of potato, where potato virus X free tips were produced by giving treatment at 35 degrees centigrade for several months. In chemical treatment, certain chemicals like antibiotics, plant growth regulators, amino acids, purine and pyrimidine analogues are added to the culture medium for virus eradication. The examples why chemical treatment has proved helpful include potato tissue cultures, where PVX has been eradicated by adding malachite green and thiouracil to the medium. Another example is of Chinese cabbage, which was produced free of TMV by adding actinomycin D to the medium. Another example is CLSV free apple and tulip breaking virus free lily by using ribavirin. Applications in plant breeding include somaclonal variations, haploid production, somatic hybridization, in vitro pollination and fertilization, and zygotic embryo culture. Variation found in somatic cells dividing mitotically in culture is known as somaclonal variation. This variation occurs through frequent genetic changes like polyploidy, aneuploidy, deletion, translocation, etc. Importance of somaclonal variations include: first, its methodology is easier than DNA recombinant technology. Second, it has proved helpful in producing disease-resistant plants like in case of rice, wheat, potato, strawberry, apple, tomato, etc. Biochemical mutants with abiotic stress tolerance have also been produced. For example, aluminum tolerance in carrot, salt tolerance in tobacco and maize. Variants with herbicide resistance have also been produced through somaclonal variations. The example of this is tobacco resistant to sulfonyl urea. Seeds with improved quality have also been produced. For example, Lathyrus Bio L 212. This is the new variety of Lathyrus sativa seeds, and these contain low content of neurotoxin. Secondary metabolites on commercial scale have also been synthesized through somaclonal variation. For example, Bio 13, which is a somaclonal variant of Citronella java, and it contains more oil and citronellone content. Another application of plant tissue culture in plant breeding is haploid production. Haploids can be produced under in vitro conditions through anther culture, ovule culture, by culturing the embryos which have only one genome. This is also known as embryo rescue. The first in vitro haploid plant production was reported by Blacksilly et al. In 1922 in case of Datura stramonium. In 1964, Guha and Maheshwari developed an anther culture technique. Haploid plants do not produce viable gametes due to the absence of one set of homologous chromosomes, so they are not able to produce viable seed set. The only way of perpetuating is by chromosome doubling in order to obtain homozygous diploids, which are known as dihaploids, which either occurs spontaneously or by chemical means like colchicine treatment. Now, importance of haploids. First is alien gene or chromosome introduction. By introducing alien genes or chromosomes in the plants, we can get high yielding varieties with disease resistance. Example of this is in case of rice, where a high yielding variety of it resistant to blasts have been produced through anther culture. Another success was achieved by releasing florin variety of wheat. Haploids, they are the tools for genetic transformation studies, like transgenic plants of Brassica napus were developed by introducing DNA into the individual pollen embryos. Haploids are also useful in the production of asexual lines of trees or perennial species. For example, Chinese produced a pollen-derived rubber tree taller by 6 meters, which was able to propagate vegetatively. Haploids are also important in inducing mutations. These mutations also help in producing disease-resistant mutants like tobacco mutants resistant to black shank disease, wheat lines resistant to scab, and herbicide mutants of Brassica have been produced through the technique of haploid production. Dihaploids can also be produced from the haploids, which have proved useful in developing new varieties of barley, brassica, rice, maize, rye, potato, pepper, and asparagus. Exclusively male plants in dioecious species have also been obtained through haploid production. Example of this is Asparagus officinalis. Another importance of plant tissue culture in plant breeding is somatic hybridization. Somatic hybridization means development of hybrid plants through the fusion of somatic protoplasts of two different plant species. These protoplasts are the wall-less naked cells. It was Carlson et al. In 1972 who first of all developed hybrid plants by fusing protoplasts of Nicotiana glauca and Nicotiana langsdorffi. Somatic hybridization involves four main steps. First, protoplast isolation. Here, protoplasts are isolated either mechanically or enzymatically. Second step is the protoplast culture. In culture, the first step is the development of cell wall, which is followed by divisions of these protoplasts to form small cell colonies. Third step is the protoplast fusion. This fusion can either occur spontaneously or they are induced to fuse by using fusogens like polyethylene glycol, sodium nitrate, calcium ions, and polyvinyl alcohol. And the final step of somatic hybridization is the selection of the final product, which are the somatic hybrids. Sometimes fusion product contains nuclear genome of only one parent due to elimination of nuclear genome of other parent. Such hybrids which contain only cytoplasmic traits of both parents are called as hybrids, and the process of their formation is known as hybridization. Importance of somatic hybridization. Somatic hybridization is very important in overcoming barriers of sexual incompatibility. For example, amphidiploid Datura inoxia plus Datura discolor and Datura inoxia plus Datura stramonium having higher scopolamine content than parent formers have been produced through somatic hybridization. After this, many interspecific and intergeneric hybrids have been produced. Interspecific hybrids were produced in case of Brassica, Citrus, Lycopersicon, Medicago, Nicotiana, Solanum and Oryza species, while as intergeneric hybrids were produced between Arabidopsis and Brassica, Brassica and Barbaria, Datura and Atropa, etc. Another importance of somatic hybridization is the production of fertile diploids and polyploids in case of sterile plants. For example, hybrids of practical breeding value were produced by fusing isolated protoplasts of dihaploid potato clone and Solanum brevidens. Another example is of rice cultivars. Stress tolerant genes can also be introduced through the technique of somatic hybridization. For example, genes conferring tolerance for cold, frost and salt were introduced in tomato. Another importance of somatic hybridization is transfer of cytoplasmic traits from one species to another. For example, a line which contained the chromosomes of only Parthenocissus but exhibited some of the cytoplasmic properties of Petunia was produced by fusing the protoplasts of these two genera. Later on, this technique was used for Nicotiana, Brassica and Raphanus plants. Another importance of plant tissue culture in plant breeding is in vitro pollination and fertilization. In vitro pollination, style or part of it can be excised and pollen grains are either placed on the cut surface of the ovary or transferred through a hole in the wall of ovary. This type of in vitro pollination is known as intra-ovarian pollination. Another approach is the direct pollination of cultured ovules, which is known as in vitro ovular pollination, and the direct pollination of excised ovules together with placenta, which is known as in vitro placental pollination. In vitro fertilization involves fusion of male and female gametes isolated from higher plants and subsequent regeneration of the fusion product, which is a zygote, into embryo and finally a plant. In vitro pollination and fertilization is important in first inbreeding. For example, hybrids were produced between Petunia axillaris and Petunia hybrida to get seed set 5 to 10 times greater than that in vivo. Another importance is interspecific, intergeneric and interfamiliar hybridization. For example, Melandrium album cross Melandrium rubrum, Melandrium album cross Viscaria vulgaris, Melandrium album cross Silene shapta and Nicotiana elata cross Nicotiana debni. After that, fertile hybrids were produced by in vitro pollination of Nicotiana tabacum with the pollen of Nicotiana rustica, followed by hybrid ovule culture. In vitro pollination and fertilization is also important in haploid production. For example, in case of Mimulus luteus, by pollinating its exposed ovules with the pollen of Torrenia forneri. Heat stress tolerant plants can also be produced through the technique of in vitro pollination and fertilization. For example, in maize at 38 degrees centigrade. Another application of tissue culture in plant breeding is zygotic embryo culture. Zygotic embryo culture means aseptic isolation and growth of sexually produced embryos with the objective of obtaining viable seeds. This culture was started in Cochlearia and Raphanus by Henning in 1904. Zygotic embryo culture is applicable in first obtaining rare hybrids. For example, Lilium henry cross Lilium regale, Lilium speciosum album cross Lilium auratum, Hordeum cross Kale, Hordeum cross triticum. These hybrids both interspecific and intergeneric, have been produced through the technique of zygotic embryo culture. Hybrids can also be produced through zygotic embryo culture, for example, by culturing the excised hybrid embryo of Hordeum vulgare and Hordeum bulbosum. Breeding cycle can also be shortened. For example, in Cyrus, the breeding cycle was shortened from 3 years to less than 1 year, and in case of Rosa, it was made to form 2 generations in a single year. Zygotic embryo culture is also important for rapid seed viability test. This has proved successful in case of peach. Rare plants can also be propagated through zygotic embryo culture. For example, Musa bulbiciana, which is a wild relative of cultivated banana, have been propagated through this technique. Another example is of Macapuno coconut. Now, the general applications of plant tissue culture. It includes germplasm conservation and secondary metabolite production. Germplasm refers to the sum total of all the genes present in a crop and its related species. A global organization known as International Board of Plant Genetic Resources, that is IBPGR, has been established for germplasm conservation, which provides necessary support for collection, conservation and utilization of plant genetic resources throughout the world. The germplasm is preserved by 2 ways. First, slow growth systems, and second, cryopreservation. In slow growth system, cultures are grown on modified cultural conditions. These modifications include reduction in temperature or manipulation of chemical constituents of growth media by using abscisic acid or high sucrose content. Slow growth systems are generally applicable for medium-term storage. This has proved successful in case of Beta vulgaris, Coffea arabica, Colocasia esculenta, Allium sativum, etc. Cryopreservation. The principle involves bringing the cultures to a non-dividing state by subjecting them to super low temperature in presence or absence of cryoprotectants, for example, glycerol, DMSO, PEG, and avoiding formation of intracellular crystal formation. Cryopreservation involves freezing of plant material by maintaining the temperature of liquid nitrogen, which is minus 196 degrees centigrade or minus 150 degrees centigrade. Cryopreservation has following applications. First, long-term preservation of cell cultures which possess the capacity of synthesis and accumulation of secondary metabolites. For example, cell cultures of Catharus roseus, Daucus carota, Digitalis lanata, Dioscorea deltoides, etc. It is also important in maintenance of stock cultures and samples that are meant for future use. For example, in case of cassava, pea, chickpea, rice, wheat, peanut, coconut, strawberry, oil palm and sugarcane. Almost an infinite number of replicates can be stored in vitro using little space, in contrast to the thousands of hectares of land that would be required for the same number of plants if maintained in situ. Through germplasm conservation, it is possible to conserve cell lines producing medicines, for example, Datura insignis, Lichnophora ericoides, Solanum martianum, etc. Genetically vulnerable plants can also be conserved, for example, citrus species and Phoenix species. Germplasm banks can also be established. Some of the germplasm banks are CIAT, CIP, IITA, IVIA, NCGR, etc. Secondary metabolite production. Higher plants are valuable sources of industrially important natural products, which include flavors, fragrances, essential oils, sweeteners, antimicrobials, pharmaceuticals, etc. In most cases, these chemical compounds belong to a metabolic group collectively referred to as secondary metabolites. Many of these secondary products, especially various alkaloids, are of immense use in medicine. For example, chiconium is a dye which is produced by the cells of Lithospermum erythrorhizon on a commercial scale. Besides, there are a number of secondary metabolite products that are being widely used for various purposes. For example, vincristine is used as anti-cancer agent. Digoxin controls cardiovascular disorders. Artemisinin and quinine have anti-malaria property. Pyrethrin is an insecticide. Morphine a sedative, and Taxol is anti-carcinogenic. So friends, I hope you might have understood various applications of plant tissue culture. See you next time with another topic. Till then, goodbye.